My name is Imogene Wells. You call me Immy. I'm a fourth-year PhD candidate in the Department of Ecology and Evolution at Stony Brook. And today I'll be showing my research from the last almost 4 years here at Brookhaven National Lab on the generalist dietary dynamics in small mammal communities in the central Pine Barrens. All right. And to go over our roadmap, so what I will be talking with you today, I'll open with an introduction to my favorite organism, the white-footed mouse, Peromyscus leucopus, and review their life history and what makes them a generalist species. And you can't talk about mice and small mammals without talking about masts. I'll be sharing with you current research on the mysteries of the mast. I'll also share with you my project design, so what I've been doing since 2021, including some information about the study site, so where we are today. Share with you my sampling protocols, because we are sampling both from small mammals and the resources that they may be consuming. And I'll share some preliminary results with you. So results from our trapping efforts, looking at the community composition and abundance of small mammals in the Pine Barrens. And also, I'm also a stable isotope ecologist, and I'll be sharing with you the isotopic niche breadth and seasonal overlap between populations of mice in my transects. So why the white-footed mouse? Usually when we think about mice, they're pests. We're not big fans of them. They're ubiquitous across North America, especially here on the East Coast, found in a variety of environments, but they are great indicators of change in ecosystem health because of this. They're found in many ecosystems, some that are disturbed, some that are pristine, and they do well regardless. So I would call them an industry species, and industry species have very important roles in ecosystems. They are important prey sources for higher trophic levels, so anything from predatory birds and larger mammals. They're also very important disease vectors for those who are concerned about Lyme disease and bacterium Borrelia burgdorferi. They are great reservoir hosts for Lyme disease, so they're also important for that. And they consume a wide variety of resources because they are found in so many different environments. They're great at consuming a plethora of resources. And I'm interested in their dietary niche, so the things that they're eating. But of course, the niche, as we know, is also comprised of abiotic factors like their pH requirements, their phenology, so when they're reproducing in their life history, what they're eating and when, temperature requirements, their nocturnal species when they're active, things like that. That's all comprised in their niche. So how am I measuring their diet without doing anything invasive? So stable isotopes are great. They're a non-invasive tool that we can use. And I'll be talking about the resource use that gets recorded in their scat and their fur. And these are two great samples to use because not only are they easy to sample from, you can repeatedly sample from your recaptured individuals and track them through time, but scat records resource use at a very short interval, so what those animals have been eating for the last few days, versus fur, which actually records resource use, homogenizes it, means it out over the last month of use. So you can use two different time scales here and look at the resource use over time. And a generalist. So what is a generalist species? They are very tolerant. They consume a wide variety of resources, and that makes them very resilient to change. Even white-footed mice even made the cover of Evolution at one point. And when we think of generalists, we usually assume that there are many individuals in a population or groups of individuals that consume a wide variety of resources across the board. But you can actually see something called individual specialization, which I find super cool. And this is the phenomena in which individuals are actually individual specialists. So you have one individual that's really good at consuming one type of resource, and their neighbor is really good at consuming a completely different type of resource that's also found in the same environment. So this is something I'm interested in measuring as well. So the degree of specialization that individuals may exhibit. And this can be dependent on time. So I'm often binning my data in terms of the season, so spring, summer, fall, and this may also depend on what where the population is. So the habitat that they're in, so white pine, pitch pine, and another type that I'm not measuring is red maple. So going back to white-footed mice, what do we think about with white-footed mice? What are they eating? Of course, it's acorns. Acorns are such an important resource for many small mammals, and this comes back to the mast. So Who's familiar with a mast? Mast cycles. Okay, great. So if you don't know masts, there are these interannual resource pulses of really big quality. So every few years, you get a high quality mast, a big drop of acorns, and that can be a really big driver for population abundance of small mammals that consume them. And they're really mysterious. We still don't understand what determines when and what quality of masts occur every single year. But what we do know is that there's a really tight positive correlation between the quality of a mast in the autumn and the population density or abundance of small mammals in the following summer. And this is a very important relationship and drives the boom-bust cycles of small mammals. And from an abiotic lens, more current research is focusing on what climatic variables drive mast cycles. So Koenig et al. Found that it's actually the conditions that happen in the spring that can determine the success of pollen and fertilization of masting crops like oak trees. And that will determine if there's a high or low fertilization success in resulting small or large, poor and good quality masts. So going back to what I've been doing for the last few years, I'm interested in quantifying small mammal communities, specifically the white-footed mouse diet, and looking at their dietary dynamics over time in space in the Pine Barrens at Brookhaven National Laboratory. And a bit about this project. So my transects are right outside in two different forest types, the white pines and the pitch pines. This is one of my trap sites. So I use Sherman traps covered with trap covers. They're all labeled with flags so we can go out and find them again. There are 16 traps per transect. They're 14 meters apart. And this allows for maximizing their home ranges. So usually our recaptured individuals, usually we'll find one that visits the same site over and over, but this can, males have larger home ranges. We've seen even up to 4 traps that an individual can visit. So it does vary with their home range. And a bit about the history of the site. Everything I've learned from Tim about the wonderful history of Brookhaven National Lab. So what's important with the white pines and the pitch pines is that what would become Brookhaven National Lab was once Camp Upton, and there were a great transformation of the landscape. What would become the white pines were then planted by the Civilian Conservation Corps in the 1930s. So the white pines, it's contested if they're actually were once on the eastern seaboard at one point or not, but this forest was planted artificially and introduced. And then the rest of the landscape, once Civilian Conservation Corps came in, planted the white pines, and everything else just was left to recover, and that became the Pine Barrens. And Brookhaven National Lab actually comprises 5% of the central Pine Barrens, and it's just a small bit of this wonderful landscape that we live in. So what does it look like when we go out in the field, me and my team of undergrads? We approach our traps. We see if an individual has visited it. If there's no animal, we still collect the scat because that has important information at the population level, regardless of the individual that deposited it. We scruff them, we identify them to sex, we take a sample of their scat and their fur, we give them an ear tag, we have a lot of great recaptures, so we record that if they've visited before. We also take some measurements, so their mass and length measurements, hindfoot, tail length, total body length, which is pretty standard in small mammal trapping. We follow the protocol outlined by the National Ecological Observatory Network, but this is really standard stuff. We also record information related to their life history. So lactating and pregnant females. We have this female that is lactating and pregnant. This female is just pregnant. We also record ectoparasite status, not formally, but we put it in our notes. So often this is the form of a botfly, tick load, fleas as well. Interesting about white-footed mice is that they typically molt twice a year, once after they reproduce for the first time in the spring, and then once before they overwinter, and also when they transition from being a juvenile to an adult. So you can see a difference in the pelage here of the rusty orange color of an adult and the gray color of a juvenile. And then this is an example of our fur sampling. You can see that this is a fresh cut of fur right from the rump, and then this recaptured individual has grown back their fur since the first sample. But we try to sample as frequently as we can. Scat is very easy, you just collect it from the body. And we resample for once there's been ample time for that to grow back. And I'm also interested in what these animals are eating, so I need to collect that. So for the first 2 years of this project, 2021, 2022, we had pitfall traps set out with mesh netting so that small animals wouldn't fall into them. And this was a great way of collecting invertebrates and seeds. Also, I've done a lot of surveys, fungi surveys, cone surveys. We've had some seed baskets out for 3 years. They weren't as successful as the pitfall traps for collecting seeds, but in the end, we sort through them and collect those dietary items for our analyses. And as a side note, I love being in the field, and I love bringing undergrads out with me. It's a great educational experience for them. We see some cool things we've seen, very large slugs. We have had some camera traps, not only to monitor disturbance, but part of an undergrad's project. A lot of white-tailed deer, no surprise. Disturbance from raccoons, having to trap them and relocate them. That's always fun. Some box turtles as well. And then, I believe this was 2022, we had an individual that gave birth in a trap. So we got to see some pups, which is exciting and a great experience to have. So using this longitudinal trapping data, what are the spatio-temporal dynamics of the small mammal community? Just using our trapping success. And we've had 3,544 trap nights since September of 2021. So trap night is setting one trap for one trapping session. So you could set 60 traps in one night and have 60 trap nights, or set one trap for 60 nights and have 60 trap nights. So this is just a measure of our effort. And we've had 359 capture events, of which there have been 274 events of capturing white-footed mouse with 179 unique individuals, so that accounts for our recaptures, and 55 have been captured at least once, some being caught as many as 8 times. There's also been a lot of other species that we'll go into as well. So I'll first share with you catch per unit effort. So the catch per unit effort means the number of individuals caught per traps set. And I'll break this down by species and also by forest type. So in 2021, we began in September, ended in October, quick season. And we actually saw the highest abundance in the transects. There was actually a crash after. We haven't been capturing masting data just observationally, but we didn't hit a mast until this fall. So this is actually in the absence of a mast. So we do see a bit of a crash in abundance here in 2022 for our mice. Oops. And in 2023, we saw something different. We saw a bit of a higher abundance upkick in the late spring, as opposed to the previous 2 years where it was more in the late summer, early fall. We can also use the minimum number of alive, which is a similar indirect measure of abundance. And this accounts for not only the individuals that you catch on a certain day, but those that are assumed to be alive because you have caught them before and after, but you just didn't catch them on that day. And this is in agreement with the catch per unit effort indices as well. We see in 2021, high abundance in early fall, 2022, higher abundance in early summer, late summer, and then 2023, higher abundance in the late spring. We've also gotten a few eastern chipmunks, none in our first year, some in our second year, and in 2023 even more. And we see they are mostly occurring in the early summer, late summer. We've also gotten some mast shrews, which is really exciting. Very small, fun to work with. Some have been alive as well. We saw them in the late summer through late fall. Also, short-tailed shrews. Every year we've gotten them, mostly confined to early fall, but laying low throughout the year as well. And my favorite, flying squirrels. They are so cool. None in our first year. Some in the second and third years, mostly earlier in the year, in the late spring, early summer. They're the most active. We see some abundance there. So what can we glean from this? There's definitely some temporal partitioning of the relative abundance of these different species in the Pine Barrens throughout the seasons, when we're catching them and when we're not. Keep in mind that the chipmunk is a diurnal species, and we're trapping overnight, so that might not be too reliable, but there's definitely differences in when we're detecting them. All right, I'm also interested in quantifying body condition, which we've seen in a few other talks before. And this I'm calculating using standard methods, which is the residuals from a regression of mass on length. So we're using one of our linear lengths, our tail length, and our weight. And individuals above this line are fat for their length, and those that are below are skinny for their length. And this is pretty standard and is a pretty good proxy of other nutritional stresses like fat deposits, et cetera. So what do we find between the forest types? Those individual mice in the pitch pine forests have consistently higher body condition, so better body condition, than those in the white pine. So those in the native forests are healthier than those in the introduced forest. However, there's no significant differences across the seasons when we break it down, but consistently, those in the pitch pine have better body condition than those in the white pine. So abundances vary spatio-temporally in the small mammal community. But going back to our other question, how do white-footed mouse niche dynamics change over time and space? And I'll be saying things like niche breadth, which refers to the width of the niche, and niche overlap, which refers to the similarity of the niche either between seasons or between habitats. And again, stable isotopes, great non-invasive tool that allows for multiple repeated samples of resource use from an individual over time. And without going into detail, I'll be using two axes, standard for ecological research with stable isotopes, looking at delta 15N, so the relative amount of light to heavy nitrogen. So a higher value is associated with an individual consuming at a higher trophic level, and that of a lower value is associated with the individual feeding at a lower trophic level. And usually this is referring to different types of photosynthetic pathways, but we're in a C3 dominated system. So our carbon values, a smaller carbon value is associated with having a less varied diet, and a greater carbon value is associated with having a more varied diet. And as you can see, this is incremental as you increase trophic levels. And going back to my figures before, this question of, do we see true generalists, different individuals that are all consuming a wide variety of resources over time, or are we seeing individuals that are consuming very specific things in their habitats? And again, we will be putting this in a biplot of carbon and nitrogen to measure resource use. And again, this can vary looking at the time of the year or the habitat. So walking through this, we have our carbon and nitrogen on our X and Y axes, breaking it down by bin seasons. What are we looking at here? So I'll just use an example of the dietary niche of the measured by the scat and the individuals in the pitch pine. So this data is actually a subset of our data because we've been collecting so much. So this is only from our recaptures, and it's 161 samples from 43 individuals. So in late spring, we're measuring a dietary niche using these standard ellipses of a niche size of 14.67 per mil squared. We see an increase in niche size to 27.01 per mil squared, and there's about a 33% overlap between early summer and late spring. In the late summer, it grows even more to 37.05 per mil squared, with an even more similar overlap with the previous season. And then in the early fall, a slight decrease in niche size to 26 per mil squared, with an even larger overlap with the previous season. Excuse me. So what are we seeing here? The smallest dietary niche is occurring in the late spring. The greatest dietary niche is occurring in the late summer. And we're seeing the most change between seasons in the early summer, late spring transition, and the least dietary change between the late summer, early fall transition. And a different way of looking at this is across forests. So now I'll be comparing the white pines to the pitch pines. And this is using a probabilistic model, a Bayesian model. And these are outputs from running that model. So you'll be seeing, I'll be highlighting the medians of those draws, but this is why it's in the form of a distribution. So again, we're comparing the two forest types in this case. In the late spring, we're seeing very different niches, so little overlap between those two forest types, so indicative of a less similar niche. And as the seasons go on, we get later in the year, we see increasing similarity between the forest types and what those individuals are consuming, and there's more overlap between the dietary niches of these populations. A little bit of a regression back in the early fall. But what can we say about this data? Populations in different forest types are the most different in the late spring, and they're most similar in the late summer. So which processes are underlying the small distinct niches in the spring and the large similar niches in the summer? Well, going back to our abundance data, we can see that those dietary niches are becoming the largest when populations are the most dense, there's the most abundance. So we can infer that as intraspecific competition, so within species competition, is increasing in the late summer of 2021 and 2022. The dietary niche breadth is expanding at the population level. So that data I showed you is only from our first 2 years of data, so 2023 is not included, and that's a bit of an anomaly with our population abundance metric. So if I was to hypothesize what I would see next, I would say that I would expect to see increased breadth and overlap occurring earlier in the late spring of 2023. And the question of, are we seeing individual change over time? So breaking this down by carbon and nitrogen, if we see an individual that's resampled multiple times with little variation, we can assume that that individual is specializing on something and it's going back to the same item over time. But if we see change, that could indicate that the individual is consuming different things over time. So what do we see here across forest types? We do see variation, both in what's captured in scat and fur. Individuals do have variation between resamples, so that's indicating that they are consuming different items between samples. And ideally, I would be using a different model for this. I'm still in the process of making that and running more samples for it. So this is the best I can give you right now. And then looking at nitrogen, same story. Lots of variation, sometimes between resamples, both increase and decrease, which indicates that we are seeing individuals that may be true generalists, or some that could be more of a specialist. So we may be seeing a mixed bag, and that's something that our next model could tell us. So in this very dynamic small mammal community, white-footed mice are expanding their population-level dietary niche in the late summer and early fall in response to that increasing intraspecific competition. But we can also see differences between forest types, and that mice in different forest types are occupying very distinct dietary niches that do reflect their different habitats. And with that, I would like to thank you for hosting and also acknowledge my collaborators and people who have helped this research along the way, especially the folks in my department at Stony Brook University. This research was paid for by a Cost of Education Award and my PI, Tara Smiley's startup funds. Also, Greg Hengus in the Hengus Lab in the Department of Geosciences for the analytical aspects of this research. And then also Brookhaven National Laboratory Environmental Protection Division and Tim Green for being our host for many years now and everything related to administration and getting this research up and running, big help. And the undergraduates that have stayed on this project for multiple years now, some are on their third year. My contact info is here if you have any questions, or you could ask me some right now, so thank you.
We do have time for questions, and I have the first question. Difference between your white pine and pitch pine areas. Are you seeing anything based on the understory?
Oh yes, oh yeah. So going back to the pictures, actually, the understories are really distinct in these two habitats. So I would think plant species composition may be pretty similar, especially because of this heath. So we see a ton more like Vicinium, so huckleberry blueberry species, coating the floor in the pitch pines, very dense. Versus that in the white pines, you do still see that, but for the mass majority, it's just saplings of white pines and bare needle ground cover. So it definitely is distinctly different and a lot more sparse cover. So that could have implications for not only what they're consuming, but thinking about behavioral changes associated with decreased cover, ability to find refuge when being pursued by a predator, things like that. So that, that aspect of fear could also influence what they're eating and their health overall.
Okay, and then a second question, we're starting to see a lot of mortality in pitch pine from the southern pine beetle. Are you picking that up in the in your research?
So I think that's a question for more this year. So it wasn't that bad in the past 2 years, so 2022 and 2023. But having gone out there for the first time this year, this week, it is becoming increasingly more upsetting to see the landscape, and which I think will be interesting following a mast year, how those two will interact. So having a more depauperate landscape, but then also having that resource pulse happen. But yeah, we'll see. Yeah.
Hi, Amy, great job. I'm too short for this. Sorry. So I was wondering, and I'm actually kind of surprised we've never talked about this because I know you use the traps, but when I did that one mammal project I did years ago and we were trapping the tree shrews, there was an issue because the tree shrews would learn that if they would learn to avoid traps because they were like, oh, I do love the pineapple and the banana that we were baiting with, but they were like, but if I get that, I get trapped. So but tree shrews I think are known to be pretty smart mammals. I don't know anything about the mammals you're working with and their level of intelligence. The reptiles I work with are not that smart. But I'm wondering if you have seen any patterns that might indicate that they are learning to avoid traps, or like if you, like, is there any kind of like sex ratio, but like sex bias in like what you're catching and like what that mean, or like do you catch more juveniles because maybe the adults are learning faster, or like, or are they not learning at all? Like do they just have no conception that they're just being trapped every time they go in these?
So okay, so I'll answer that in two parts. So in comparison to like bait type, we use oats instead of what people usually use for small mammals, which is peanut butter, because that's a lot more attractive and a lot more tasty for them. But because we're using, we will be using a mixing model to determine exactly what resources they're using. We don't want to introduce peanut butter because that is such a strong signal. It could really attract them, and it might, knowing what oats are in this model will be easier than using peanut butter because it's very enriched in carbon too, which can really skew what we're getting out of that model and identifying resources. So that's the reason why we don't use something that is super tasty for them. We do see so our recapture rate is consistently about like 33%, so we get about 1/3 of those individuals back consistently. I don't think they're really attracted to the trap sites, maybe because we're using not a super attractive bait. Would they be going and visiting the traps knowing that there is a food source there? Potentially, yes. But yeah, that's yeah.
Okay, cool. Thank you.
So this might be another topic for another year, but did you encounter any meadow jumping mice or no?
No meadow jumping mouse, no voles. We are permitted to trap, live trap, all the list of small mammals that are known or thought to have occurred at Brookhaven National Lab, including those more uncommon species. Never seen them. So we only started to see the masked shrew and the short-tailed shrew. I mean, even short-tailed shrew is pretty common, but masked shrew we did see for the last 2 years, and like that was a big deal to us. We're not seeing anything more rare than that.
Oh, thanks. Great presentation. Thank you very much. Just a quick question: are there any concerns ever when you're sampling in regard to hantavirus exposure?
Yes, of course. So because we did start about in the, still in COVID era, 2021, I would make some jokes about that. Like, we're already wearing the N95s, like, no risk of hantavirus. Good thing we have our masks on. But yes, we're very, we're aware of the risks involved with sampling from small mammal scat. We always make sure that we're not, breathing into our traps, always kind of at an angle, making sure that we're holding our mice at a safe distance, and we're reducing that risk, because that's definitely can be a problem, yes.
Thank you.
Another quick question for you over here. I know there's some evidence, and I think maybe, Tim, you've documented with the tiger salamander study about shrews feeding on tiger salamanders. Is there any evidence of white-footed mice feeding on salamanders or amphibians in your study? Did you find any evidence of that?
I'm not including amphibians in my diet model that I'm working on, mostly because I'm not in any kind of wetland, so it's not something I hadn't really considered, other than more like moth and beetle larvae or other invertebrates, but I'm not putting any amphibians. I hadn't considered amphibians for these habitats. Yeah, that I could legally sample too, and use for this model.
All right, thanks, Emmy.
Thank you.